Good morning, students. Welcome to Bindu Dadwal Chemistry Lessons. Our topic for today is structure of RNA. The structure of RNA is similar to that of DNA, except that it is a single strand structure. In RNA, there is only a single strand consisting of sugar molecules, phosphate, and nitrogenous base. Only one strand. Whereas in case of DNA, it was a double-stranded structure. RNA molecules are of three types. They perform different functions. They are named as messenger RNA or mRNA, ribosomal RNA or rRNA, and transfer RNA or tRNA. Biological functions of nucleic acids. DNA is the chemical basis of hereditary and may be regarded as the reserve of genetic information. DNA is exclusively responsible for maintaining the identity of different species of organisms over millions of years. Important functions of nucleic acids are replication, transcription, and translation. Replication is the property of a molecule to synthesize another molecule. So DNA has the unique property to duplicate or replicate itself. It is an enzyme catalyzed process. In this, the two strands of DNA open up. Each strand will act as a template for the daughter strand to be built up. The specificity of the pairs to attach with each other, for example, with adenine only thymine will attach, with cytosine only guanine, with guanine only cytosine. This helps in the synthesis of DNA. So at the time of cell division, the two strands of the DNA will open up and each strand will serve as a template for the synthesis of new DNA strand. So when it opens up, the complementary bases, nitrogenous bases from the cytoplasm will come in front of the strand. For example, a cytosine will come in front of guanine, thymine in front of adenine, and then they'll get joined together, and as a result the daughter strand will be built. So due to unique specificity of base pairing, the newly synthesized complementary strand in each core is an exact copy of the original separated from it. As a result, two double-stranded DNA molecules are formed called two daughter DNA molecules. One of the strand comes from parent DNA molecule and other is newly synthesized. Each DNA is exact replica of the parent. In this way, the hereditary effects are transmitted from one cell to another. It may be noted that DNA replication follows the base pairing rules by which adenine pairs with thymine, guanine with cytosine. This can be shown here. These are the two strands of a DNA. These will open up and one strand will act as a template for building up of another strand. So these are the two strands here. So in front of A, T will come, and as a result a new daughter strand will be built up. T in front of A, C in front of G, G in front of C, A in front of T, and T in front of A. So we get another strand. Similarly, the second strand will act as a template for building up a new daughter strand, and after the formation we see that the two DNA molecules are exactly similar, identical to each other, and as a result the hereditary characters are passed on from one to another without a change in itself. The DNA replication is semi-conservative. Only half of the parental DNA strand is conserved and another half is synthesized. Replication always begins from the 5' direction towards 3' direction. The second process function of DNA is protein synthesis. DNA molecule will perform the function of synthesizing proteins which serve as a machinery of the living cells. In this process, the genetic information coded in the form of specific base sequences is translated and expressed in the form of sequence of amino acid, which result in the synthesis of specific proteins which perform various functions in the body. These amino acids will be built one after the other to build the overall structure of protein. So, genetic information coded in the form of specific base sequences is translated and expressed in the form of sequence of amino acids, which result in the synthesis of specific proteins which perform various functions in the cell. Actually, the proteins are synthesized by various RNA molecules in the cell, but message for the synthesis of a particular protein is coded in the DNA. Protein synthesis is a fast process and about 20 amino acids can be added in 1 second. For example, silk has a major component, fibroin protein. A single fibroin gene can make 10⁴ copies of its messenger RNA, and each messenger RNA can produce 10⁵ molecules of fibroin protein in 4 days. So in all, a 10⁹ molecules of protein fibroin can be constructed per cell in a period of 4 days. Difference between DNA and RNA. So DNA occurs mainly in the nucleus of cell. RNA occurs in the cytoplasm of cell. DNA has a double-stranded alpha helix structure in which 2 strands are coiled spirally in opposite direction. RNA has single strand alpha helix structure. The sugar molecule is 2-deoxyribose in DNA. It is ribose in RNA. Nitrogenous base uracil is not present. Nitrogenous base thymine is not present in RNA. DNA has unique property of replication. RNA does not replicate. DNA is responsible for transmission of hereditary characters. RNA helps in protein synthesis. DNA molecules are very large. Their molecular mass ranges from 6 × 10⁶ to 16 × 10⁶ units, whereas RNA molecules are much smaller with molecular mass ranging from 2 × 10⁴ to 4 × 10⁴ units. There are so many millions of base pairs in each person's DNA that every person has a different sequence, and this technique of identifying individual person based upon the uniqueness of their DNA pattern is called DNA fingerprinting. It is the same for every cell and cannot be altered by any known treatment. Each segment of a DNA molecule that codes for a specific protein or polypeptide is called a gene, and the relation between the nucleotide triplets and amino acids is called the genetic code. The chemical change in the sequence of nitrogenous bases in a DNA molecule that could lead to the synthesis of protein with different amino acid sequences is called mutation. That's all in today's video. If you like the video, please do share and subscribe. Thank you. Have a nice day.